Really excited to be here with Dr. Mariana Petra Hanga, also known as Petra Hanga. She's a lecturer in biological engineering at Aston University, and we're really proud to have her as a member of the GFI grantee family. I believe she was part of our inaugural cohort of grantees. She's a biochemical engineer by training, and she's really interested in this intersection of engineering and the life sciences, which you'll really see play out during her seminar. And her research focuses on two distinct grand challenges. The first is on the healthcare side. Can we develop future therapies that will minimize the economic and social burden of an aging population? And then the second is building better food and nutrition for health by advancing cultivated meat technologies that will help us kind of shift away from industrial animal agriculture. So we're really excited to have her here today. I'll mention that she recently launched a publication that covers a lot of the research that she'll be talking about today. I just dropped the link there in the chat. It was published in Biotechnology and Bioengineering Journal and covers this bioprocess development for the scalable production of cultivated meat. So I will kind of finish talking now. Again, very excited to have you all here and very excited to have you here, Petra. So whenever you're ready, feel free to take it away.
Thank you so much, Amy. This is absolutely fantastic. I'm really excited to, first of all, to have this opportunity to give the seminar and to present my work as part of the project that has been funded by the Good Food Institute. So thank you so much, everyone, for joining me, and hopefully you will find this as exciting as I do. So I thought about, starting by saying who we are, first of all, because I bet that not many of you have heard of Aston University. We are a university based in Birmingham, United Kingdom. And we're actually right in the heart of Birmingham. Birmingham is, just as an extra topic, Birmingham is the second largest city in UK. So this is us. Before we actually started working on the Good Food Institute funded project, actually doing some work in production of cultivated meat, we've done a lot of work on human cells, and we've worked with a variety of stem cells in particularly. So we looked at human mesenchymal stem cells, human embryonic stem cells, induced pluripotent stem cells, even olfactory in-sheeting cells. So we've done bioprocessing, not only covering the expansion of those cells, so the upstream, but also looking at different technologies of different ways that we could improve the downstream when working with cells for purpose of cell therapies. We're referring mostly to things like how do we harvest those cells in a manner that we can actually still retain the properties and the qualities of those cells that makes them useful for therapies. So in our bioprocessing experience, we've worked with a range of bioreactors. All of these cells are adherent-dependent cells, so essentially they need a surface to attach to. So when you're growing these cells in suspension in bioreactors, you still need that surface. And that surface is essentially offered by microcarriers, which you can see here in the right-hand side picture. So the green dots that you see there, those are essentially live cells that were growing and populating those microcarriers in suspension. So as I've mentioned, we've worked with a range of bioreactors from the 15 milliliter tic-tac boxes, shaped bioreactors from the Amber 15 system from Sartorius to 250 mils to all the way to the Litre scale bioreactor vessels. But as I said, these were all, so all the experience that we've had was with human cells. And these are just some publications. So essentially, as I said, we weren't just looking, oh, I think my camera has gone off. Can you still hear me? Oh.
Yes, we can hear you.
Okay, sorry about that. Yeah, okay, it's back on now. So all these publications are looking at not only, as I said, not just the upstream, so the expansion in itself, but we've also done research into what is known as the bead to bead phenomenon, which essentially occurs when you add additional surface area to in your bioreactor. Your cells will start jumping from one microcarrier to another to populate that fresh surface area. So we've done investigations in that. We've even looked at novel type of surfaces that we could use inside the bioreactor. So the concept of a temporary liquid microcarrier made of a perfluorocarbon, or the concept of a temperature responsive microcarrier. And the advantage of those type of surfaces is not that they would promote, not just that they would promote cell expansion, but they would also help with the harvesting of the cells at the end of the culture, and very importantly, non-enzymatic harvest. Okay, so there have been loads of studies where proteolytic enzymes, which is the typical method of harvesting cells, and this is also applicable to bioreactors, to microcarriers. They have been proven to have detrimental effects, particularly on the cell quality. And this is important if you're working with cells for purposes like therapies, because you know, at the end of your culture, you still want those cells to have those critical quality attributes that makes them useful. So in 2018, we were lucky enough to be, you know, awarded by the Good Food Institute and be one of their grantees, which essentially, you know, opened up all of our interest in cultivated meat. And we've seen, you know, we've seen the possibility of applying all these skills that we have in bioprocessing and in working with stem cells in a new field that, you know, undeniably has some very real and very significant, well, will have and has some very real and significant benefits. So as part of our, well, as an outcome of the GFI project, we've actually published, as Amy mentioned earlier, our first paper from this project not long ago, a couple of months ago, which we are very proud. But we're also working towards a second paper, and a lot of the data that we'll show today essentially focuses on that. Yeah, we'll go into the publication that we are currently preparing. So when we started working on this project, as I said, all the experience that we had was with human cells. So we had to make a lot of assumptions that whatever works and whatever is, you know, it's out there, and whatever we know from culturing human stem cells will be directly applicable to other species like bovine. So we started doing a little brainstorming session to think about what could be a possible suitable cell source to start the production of cultivated meat. And what we came up with, there were three. So satellite cells, mesenchymal stem cells, so adult stem cells, and pluripotent stem cells. So we kind of did a bit of pros and cons for each of these type of cells, so that we can actually decide on a cell source. So with satellite cells, we know they can differentiate into muscle, but on the downside, they can only be isolated from muscle tissue. They are primary cells, which means that you will only be able to grow them for a limited number of passages or population doublings. And, you know, if you're thinking about the meat as a full-on product, it is complex, yeah? It has a very complex structure. It doesn't just have muscle cells in it. It also has fat cells. It also has connective cells that keeps everything together. So if you do decide to use satellite cells, then obviously the fat needs to come from a different source, okay? But that essentially in itself would lead to some complications in terms that you would now have two cell sources to use. You would have to essentially adapt to the culture of those two separate cell sources. And you would need to have multiple bioprocesses for the expansion and the differentiation of those cells as well. The other cell type that we thought that it could be, a potential cell source are mesenchymal stem cells. And the reason for that is because we know these cells are present in adults, yeah? We know we can isolate them from multiple tissues, bone marrow, fat, umbilical cord. And we also know they can differentiate to fat. And there have been some reports in literature about MSCs being differentiated to muscle as well, which can come in really handy because at this stage you would only have one cell source to work with in the first instance. On the disadvantages, on the cons, these are also primary cells, the same as the satellite cells. So you can only grow them for a limited amount of population doublings. And actually, those reports about muscle differentiation, there are only a few, but it's even using those media formulations is still seems like it's pretty difficult to differentiate them into muscle. It is possible, but it's not possible with all MSC lines. So that was something that we had to take into consideration. The other types, which are pluripotent stem cells, so I split them because even if they are very, very similar, yeah, they have the same properties. They can both form any type of cell, so fat, muscle, connective, whatever you actually need. In this case, all three that are required for meat. There are differences in terms that embryonic stem cells come with those ethical issues associated to them because they are isolated from embryos. And that isolation process leads to the destruction of embryos. Because of that, there's also very limited availability. And, they've also been studies, again, all this information that we have is mostly from human cells. Yeah, so we were making the assumption that is the same with other species. There's also that tumorigenicity potential of these cells, which could pose safety issues. iPSCs, so induced pluripotent stem cells, they have the same characteristics, but they come without the ethical issues. And the reason for that is because they are essentially somatic cells that have been induced to the pluripotent state. But actually making iPSCs, because you would have to make them, you can't really isolate them from somewhere. You can isolate the somatic cells, but you will have to make them. You will have to induce those cells using, possibly the Yamanaka factors. From our knowledge, there are no cell lines available yet. I know there are groups working on, establishing cell lines for all of these cell types, which is fantastic. But at the moment, there's no cell lines available except for the MSCs, which I will explain. And those are the cells that we have in the end decided to use. Okay. Right. So as I said, MSCs, we were initially aiming to get bovine bone marrow derived mesenchymal stem cells. And we were actually lucky enough when we started the project to find this company, Celida Biotech. They are based in Zaragoza, Spain. But they didn't have their bone marrow derived MSCs ready to be commercialized yet. So they were still essentially producing those cells, those lines. But they had their bovine adipose-derived stem cells, which is still a mesenchymal stem cell type. So we settled for this. We said this is good enough. Learning from what we know from human mesenchymal stem cells, because we have worked with both bone marrow derived and adipose derived. The human adipose-derived stem cells actually are slightly faster in their proliferative potential than bone marrow-derived ones, and they do tend to differentiate a lot easier towards the tissue that they are essentially extracted from. So in this case, would be fat. So based on those. That knowledge that we have from human cells, we decided to go with the bovine adipose-derived stem cells. But we've never worked with other species of stem cells, so it was all pretty new to us. And we pretty much went step by step by applying what we knew from human cells. So the medium that we've used was a serum-based. And we used alpha mem from Lonza with 1 gram per liter glucose, supplemented with 10% FPS, 2 millimolar ultraglutamine, and 1 nanogram per mil BFGF. And we kind of monitored for the first passage, for the first expansion passage, when we generated our working bank. The morphology that we got at the different time points was very, very similar to what we've seen with human cells. But these cells were actually smaller in size than what we were used to with human cells, which was absolutely fine. Because smaller most of the times means that they actually grow faster. So that was pretty good. So we knew they can grow in that media, which was really good. So the next step essentially was to establish a baseline for the culture. But obviously for the monolayer culture, because once you have that really pinned down, then you can move on to translating the expansion to bioreactors. So for the baseline, first of all, we needed to know what are the limits in which we can use these cells, and what kind of, so by the limits, I'm referring to essentially the passage number that we can use the cells until they stop growing. Yeah, they slow down and they become senescent because they are primary cells. So we've essentially cultured continuously. We've got the cells at passage 1, and we cultured them up to passage 10. And what we've noticed was that from passage 7 onwards, they slowed down their growth. We've also seen an increase in their cell size beyond passage 7 as well, passage 8. So those are signs that the cells, when the cells start getting bigger and they slow down that growth, it's a sign that they're starting to become senescent, which makes them really unusable to what we want to do. So we knew that we can grow these cells up to passage 7 with no problems, and essentially I wouldn't put them in a bioreactor beyond passage 7. Yeah. So the next step to establish that baseline was essentially to characterize the cells. And because there's no, so there aren't really guidelines on how you can characterize other species stem cells. We've just taken what we knew from human mesenchymal stem cells, and this is quite old now, but it was a position paper that it's still quite widely accepted for human mesenchymal stem cell characterization. It was written by Dominici M. et al. In 2006, and it was in collaboration with ISCT, so International Society for Cellular Therapy. And essentially, they were laying down some criteria, some minimal criteria for characterizing this type of cells. So that's exactly what we've taken forward and we've used as well. So What is that minimal criteria? Well, first of all, you need to show that the cells can differentiate towards the 3 lineages. So adipogenic fat, yeah, osteogenic bone, and chondrogenic cartilage. And then you also need to show that they do express some of the markers which are positive for mesenchymal stem stromal cells like CD73 and CD90. And then there's a bunch of other assays that you can carry out if you want to have an even stronger case for the characterization of these cells. And that also includes CFUF, so colony forming unit fibroblasts. So that gives you an idea of the clonogenicity potential of these cells. So we've done that. I have to say that for differentiating the cells into all these 3 lineages, what we've used was a commercially available media. And if you look at the adipogenic differentiation, there's very little, which is shown here by through staining with lipid tox. So essentially what you see as green, those are differentiated cells towards fat, towards adipocytes. Osteogenic worked really, really well. We can see bone mineralization formed, which is essentially shown here in black. And we can also see some alkaline phosphatase present. And then chondrogenic also worked really well because you can see the blue staining for chondrocytes, yeah, for cartilage. But with adipogenic, it didn't really work as well. And I'm going to go in a few minutes a bit more in detail in that. One thing that I do want you to note was when we looked at the clonogenicity potential, so CFUFs, you can see a significant drop when you're using cells which are quite young, yeah, so early passage 2, versus cells which are at passage 7. So again, this kind of confirmed what we've concluded previously when we were looking at the expansion over 10 consecutive passages. Another thing to bear in mind is that a lot of the antibodies that are on the market, they are for human cells. They are specific to some of the other species as well, like mouse, which are there's a higher, how can I say, market demand for those. But for bovine, we had a bit of a struggle because the only ones that we could really find and they worked with the cells were CD73 and CD90 that had reactivity for bovine cells. We couldn't really find CD105, and then I think the only other one that we could find, at least in UK, they might be available in other places of the world, but I'm not really aware of it. But in UK, using our normal providers, I think the only other one that we could find was CD34, I believe, that usually these cells should be negative for. So that was pretty interesting to see. Again, it was something new because we were working with these cells, and we automatically assumed that all this commercially differentiation media would work, but clearly it hasn't. So this is the media that we've used from StemPro, Thermo Fisher. We've used the same ones, so the osteogenic and the chondrogenic from the same brand, and those worked. But in terms of the adipogenic, it didn't really work as well as you've seen it. And I mean, what makes us think that happens there is because there's probably something in the metabolism of bovine cells that is not necessarily taken into consideration in the formulation of these type of media. So it's essentially missing somehow. So first we thought maybe they're the cells. So what we did was we went to some of the cells that we've worked with before, and we know, they've differentiated with this type of media. So we went back to a few donors of human mesenchymal stem cells, and we've also tried it with a cell line called 3T3L1, So these are essentially mouse fibroblasts that can be induced to differentiate to fat. And as you can see, it worked really, really nicely. And when we quantified it, I mean, there's definitely a significant difference here. So our conclusion was that actually the commercially available media doesn't work with these cells, with the bovine cells. So the next step was to try and see if we can improve, if we can add some things to the commercially available media so that we can improve this differentiation of the bovine cells. So we've read the literature, we went back to the literature and we started reading it. And there were studies where they were saying that if you precondition the cells with insulin first, 10 micrograms per ml insulin, and then you change the media and you use the same adipogenic medium, that will improve the differentiation. It did, so it increased it to about 38%, I believe it was. We've also looked at people using, well, supplementing some of the media, differentiation media with FBS. So this, in my knowledge, this commercially available media, it's actually serum free. So we thought, okay, let's try different concentrations of FBS in the media, see if that makes a difference. It increased it compared to StemPro, which was the base, but 1%, supplementing it with 1% was the one that worked the best compared to the other ones. But still, it was still around between 20 and 30% differentiation efficiency. So, if you want to produce fat for your cultured meat, you would definitely need to be able to differentiate, to have a higher adipogenic differentiation efficiency. So we went back to the literature again, and we found some papers where they were using, preparing essentially their own adipogenic differentiation media formulation, but this was a serum-based one. So we've used the same concentrations and we've tried that, but there were also reports where they were saying that insulin concentration is really, really important, and you really need to find the right concentration for your cells. So we did that. We've made up our own media, which was a high glucose DMEM, supplemented with glutamine, 10% FBS. We kept the BFGF there, which we know is good for proliferation of the cells. And then there were all these other components that they were using, so dexamethasone, IBMX, indomethacin, and then insulin, which seemed to be pretty critical. But we varied the concentration of insulin to see which one would give us the best adipogenic differentiation efficiency. So we've done that, and we've actually got the 10% insulin, 10 micrograms per ml insulin concentration to be the best one that yielded somewhere around 80% differentiation efficiency, which was really, really exciting. And to be honest, we weren't really expecting that. But looking, so having a closer look at the cells, what we've noticed was that as the culture has progressed, so this was done over 21 days, which are the typical adipogenic differentiation protocols for human cells. But as the culture progressed, we actually seen a lot of cell death, and the cells, they somewhat looked pretty stressed when we've used these formulations, all of them, regardless of the insulin concentration. So that was pretty interesting. I mean, we're still investigating that because of COVID. We haven't had access to our lab for a while, and now we're back in, but with very limited access. But it's definitely something that we want to find out more. Maybe, I don't know, maybe we can go back to the literature, see if there are any new reports that we can find, and that we can test these adipogenic differentiation media formulations. So it's definitely one of those still open questions that we want to address. So moving on, we know that, we knew that we can we can grow these cells in monolayer, which was good. We knew the media formulation obviously is not ideal because it still uses serum, but something that I haven't tried, well, we have tried, but I haven't shown it here because it was literally just a few images. We have tried a commercially available serum-free media for, well, again, optimized for human mesenchymal stem cells. And the bovine cells grew okay in the first passage, but then in the second passage, once they have been replated, they just didn't recover. They just didn't attach. So, you know, our conclusion at the time, this was just one media, so we haven't tried it with others from other manufacturers, but our conclusion at the time was that for now, until we have something that, you know, we're really happy with, we're just going to carry on doing all of our research using serum-based media. So the next step was to translate this to, obviously, to a scalable production. Yeah. So thinking back about, you know, the world's first burger patty from meat, this is a picture that they always share and they always show where, you know, you can see the amount of flasks that was needed just to generate enough cells that could go into that burger patty. So this, you know, this has always had an effect on me because I know, you know, I always think in my mind, we can definitely do this easier, yeah? But the way that we can do this easier is by obviously using some sort of a bioreactor. So we started with spinner flasks, which are essentially small bioreactors with a capacity of 100 milliliters volume. These are, so they don't offer the full control of a bioreactor, of a Litre scale bioreactor, because they rely on the incubator for temperature and gases, but they do have, they do give you the option of doing suspension culture, so you can have an agitated environment rather than having a static environment that you would normally have in your flasks, culture flasks. So we always use, and this, you know, we've always used these for development studies for our bioprocesses, even for human cells, and they can be quite useful. So we started with the spinner flask. Obviously, you need that surface for the cells to attach to, which is offered by microcarriers. We've made some decisions here based on our experience with human cells. So we've carried out, but we were also kind of trying in a first instance to mimic what we found in the monolayer studies of growing these cells. So we decided to go for the first couple of runs with a 5-day culture on microcarriers. The microcarriers that we used were plastic from Solo Hill, I think now is Pal Life Sciences. These are essentially polystyrene-based microcarriers, so they mimic the actual tissue culture vessels quite well because they're also made of polystyrene. The surface area that we decided to go for, and this again is based on what we know from human cells, human mesenchymal stem cells, was 500 square centimeters per spinner flask in a volume of 100 milliliters. And the initial seeding density, which is what we've used for the initial monolayer studies, and again, that number is, we've taken it from the experience with human cells, was 5,000 cells per square centimeter. So comparing the first spinner flask runs with what we've got in monolayer in terms of cell numbers. So we looked at things like fold increase, we looked at specific growth rates and doubling times. There wasn't really any significant difference between this particular bioprocess, initial bioprocess, and the one in monolayer, but we know we can do better. So one thing that we've noticed was that the cells were populating the microcarriers really, really quickly, a lot quicker than what we were used to with human mesenchymal stem cells. So that in itself told us that the seeding density that we were using was probably not optimal. So the first set of experiments that we've done in the spinner flasks were to look at the seeding density. And we've done that. So we varied the seeding density from 1,500 cells per square centimeter to 6,000 cells per square centimeter. What we've noticed was that with the 6,000 cells per square centimeter, they would grow up to day 5, after which you would have a plateau. So that in itself tells me that the cells were reaching confluency on those microcarriers. They were occupying the surface of the microcarriers very quickly within the 5 days. The 1,500 cells on the opposite, except for this point at day 9, they seem to have quite a good increase, and they were the ones that gave us the highest cell number at the end of the culture. And this was an image that we've taken for 6,000 cells per square centimeter, and it was taken at day 5. So the level of aggregation that we were getting, even at that time point, was really high, was quite high. So we believe, obviously, aggregation, when you're working with microcarriers, is an indication that the cells have pretty much started to occupy all of the surface area that's available. So by doing that, then it's only expected that you would have a plateau in cell number. So again, when looking at fold increase, specific growth rate, and doubling time over the 3 different seeding densities that we tested, it was pretty clear that 1,500 cells per square centimeter was the most, was giving us the optimal results, and it was the most significant. So for the next set of experiments, and from there onwards, we've only used 1,500 cells per square centimeter as our seeding density. We also know that culture medium is usually the main contributor to cost, especially when you're growing cells. So what we wanted to do was to look at the effect of different feeding regimes, feeding strategies onto cell growth. So we came up with several strategies. So one thing that it's worth mentioning is that when you're working with microcarriers, unless the vessel, the bioreactor vessel that you are working with, it's equipped with a special type device, you can't do a full medium exchange, that meaning 100% medium exchange. You will always have some sort of a dead volume. And the reason for that is because your cells are now sitting on the microcarriers, which are sitting at the bottom of the vessel. So you don't want to remove that, because you would be losing your cells, essentially, your product, so with this particular vessel, we can do up to 80% medium exchange. So in 100 mils, you can remove 80 mils, but you will have 20 mils left behind. So that is typically what we do with human cells, but we wanted to see how would that impact. So the next strategy was do an 80% medium exchange at day 3, which is the first time point that we've carried out the medium exchange, and then thereafter just do a 50%. So that was the 80-50. 65% where you're changing 65% of the media throughout the culture, and then 50% the same, only 50%. So looking at this as expected, 80% did the best. The reason for changing the media in your cultures is essentially there are 2 functions for that. Yes, so one of them is to add nutrients, so to replenish those nutrients that have been used by your cells during culture, and to remove the waste products. Because what we know is that if waste products accumulate at certain concentrations, which are dependent on each cell type, there will be cell growth will arrest if that concentration is reached. So medium change, so feeding regimes are actually pretty important, particularly when you're developing a bioprocess for producing loads and loads of cells. So 80% performed the best, 50% was the most performed poorly, very poorly. But there's another aspect that needs to be taken into consideration. So we know culture medium is the main contributor to cost, but then you also need to think, so there also needs to be a balance essentially between the cost and between the number of cells that you are producing, so your production cell yield. And if it would be to plot the cost, here is plotted in sterling pounds, British pounds, per each million of cells that have been produced. 80% medium exchange seems to be the most effective, the most cost effective. So, which is not really what you were expecting. I mean, when we think of cost, we just think of cost, but we don't really take into consideration other parameters. And in this case was the production yield. So 80% obviously gave the best cell yield, and it was the most cost effective. So we've decided to go with this for our next set of experiments. At the end of the culture, so I've mentioned this earlier as well, very briefly, it's very, very important that you do assess your cells' quality. Yeah, so when you grow cells, particularly in bioreactors where you have all these additional environmental factors, yeah, the mixing, and everything else is in there, you do have to make sure that at the end of your culture, your cells still have the same quality as when they were, as essentially before they were put in the bioreactor. So that's what we call post-expansion, essentially. So we've followed the ISIT criteria, again, the Dominici et al. Paper, and we differentiated the cells. They still had very little adipogenic differentiation using the StemPro. But then there was bone mineralization and a little bit of alkaline phosphatase in there. And then we could see the blue Arcean staining for chondrogenic. In terms of the CFUFs, we've harvested the cells from each of the spinner at the end of the culture so these are just representative images, but it looked pretty similar to all the conditions that we've tested in terms of the differentiation. So for CFUFs, again, we haven't found any significant difference in the number of colonies that were formed from the cells that were cultured in the different conditions. So that was very encouraging. Oh, that wasn't supposed to be there. Sorry about that. So the next thing that we've looked at, and this is actually something that we want. It's some of our most recent data, and we're currently preparing another manuscript to capture this. But another thing that you can do in when you grow cells, because we know once they run out of that surface addition, that's pretty much it. You need to stop the culture, well, before that happens, essentially, before they reach that confluency. So another thing that we've looked at was surface addition. And we started with 500 square centimeters, as previously. We had the same sitting density of 1,500 cells per square centimeter. So all those things that we've established previously, we've now incorporated them and fixed them in the development of our bioprocess. But what we did now was we topped up with an additional equal surface area. So now from 500 square centimeters, you end up having 1,000 square centimeters. But in these spinner flasks, we can only have a maximum volume of 100 mils, so we kept that constant. And also 80% medium exchange that was established from before, so we carried on with that. And this is what we've got. So baseline is essentially where is the bioprocess that I've showed previously, where you don't add any sort of microcarriers, you just carry on with the 80% medium exchange at those time points. But then what we did is initially we've added the microcarriers at day 7 in the culture, and this was based on visual assessment of the culture. So we looked at the level of aggregation. But what we saw with that was that actually there was a slight dip after we've done that in the culture, and the culture didn't really recover. So that told us that actually those microcarriers probably need to be added earlier on within the culture. So we've moved on to adding that at day 5. And actually that made quite a big difference because the cell growth picked up and it was actually higher than even the baseline. So that was really exciting. Another thing that we always do, and this is common practice in my lab, is that we pair cell count with monitoring of glucose and lactate as a medium, as a minimum. So glucose, what we know happens is that it gets consumed in culture, and then lactate gets produced once you have more cells in there. So we always try to monitor it because we know, as I've mentioned previously, that if glucose gets depleted and if lactate, so metabolites, lactate is just one of them, get accumulated in culture, those can lead to, first of all, arrest of cell growth, but then it can also affect quality of the cells. So we always monitor those two things together with the cell counts. In this case, when we were looking at the baseline and the microcarrier addition, so in this graph, I've only included the bottom graph. I've only included the microcarrier addition at day 5, which was the one that performed better. And as you can see, there wasn't really much of a difference. It was pretty constant. So there wasn't really a significant drop in glucose or a significant accumulation of lactate. So that was good. So the takeaway messages from this particular piece of work was that the surface area addition at the right time point can definitely improve cell yield. As we've monitored, there was no glucose depletion or lactate accumulation. So the next thing was, again, think about surface addition, but something that we couldn't really do previously because we were limited by the size of the spinner flask, which was only 100 mils, was to, so essentially when you top up that day 5 time point with your surface area, additional surface area, also top up, so also double the volume, okay? So what we've done, again, we started with 500 square centimeters, but then we topped up at day 5. So, oh, actually, sorry, here I forgot because I've adapted this. We're now talking about 200 milliliters working volume to start with, and then 400 milliliters at day 5. So the surface area here was actually 1,000 square centimeters to start with, and topped up with an additional 1,000 centimeters. So you end up having, after day 5, you end up having 2,000 square centimeters in there. We kept the cell seeding density of 1,500 cells per square centimeter, and we kept the 80% medium exchange. But what we did now is we used a larger spinner flask. So this has a capacity of 500 milliliters. And similarly as the previous one, it will go into the incubator, and that's how we can control temperature and essentially gases. So what we've seen here was that actually the cell number, there was no, if you remember previously when initially the microcarriers were added at the wrong time point, there was a slight dip in cell number. That wasn't the case here. So the cells actually grew pretty continuously. It was pretty quite a typical growth. From putting in 1.5 million cells, we got out something like 160 million cells. So I would say that is pretty good. So as usually with everything that we do in bioreactors, we've also monitored glucose and lactate. Those arrows are to point out day 5, when remember this time we've also topped up with additional volume, yeah? So now we've moved up to 400 milliliters. And it looks like it was pretty constant. There was no signs of glucose depletion or lactate accumulation. So plotting all of those things together, yeah, what we've seen was that when looking at the fold increase, so essentially what you've got out of the bioreactor at the end of the culture, it's pretty clear that by adding surface area combined with the volume, you get the most significant increase in cell numbers. So take-home messages: surface addition at day 5 is definitely the right point, yeah. And then improved growth if the volume is also doubled at the same time. And you have to note here that actually the fold increase now compared to the baseline is more than 2 times higher. So that was quite impressive. We didn't really know exactly what to expect, so this was really exciting. So we've done all of this work in spinner flasks. Now we wanted to move to the Litre scale. And these are some pictures of some photographs of the type of assembly that we've used. So we have our controller for the bioreactor. We've used the Mobius millipore single-use disposable vessel that this controller is adapted for. And essentially all the process takes place on the bench by using peristaltic pumps, using addition bottles, and using a sterile welder to ensure that the connections are made sterilely. So everything is done on the bench. Right, so how did we do this translation? Do you just randomly start? How do you translate from a spinner flask to a Mobius bioreactor? So I thought this would be useful to cover. So we know we've had all these parameters, we've established all of these parameters in the spinner flasks, yeah, so sitting density, 1500 cells per square centimeter, the volume was 100 milliliters, surface area was 500 square centimeters per 100, and then, excuse me, all of those things. One thing that I forgot to mention was that stirring was constant throughout most of the experiments that we've done in spinners. Excuse me. So translating this to the Litre scale, we kept the same seating density because we know that works. But now so the vessel has a maximum capacity of 3 liters. We decided to start with a volume of 1 liter. So the surface area now was essentially translated for that volume. So if before it was 500 square centimeters per 100 milliliters, now we had 5,000 square centimeters per 1 liter. Medium exchange and then everything else. So medium exchanges were kept at the same time points. And the stirring now, we visually assessed it as, so one thing that I forgot to say previously was that the stirring of 30 RPM that we used in the spinner flasks with assessed it visually as being the minimal stirring speed that is required for the suspension of the microcarriers. So we've done exactly the same thing in the bioreactor, and we found that 60 RPM is that minimal agitation speed. The only thing that we could not really translate was the medium exchange. So, and I'll explain why. As I said, even in these Litre scale bioreactors, unless you have a specific device based on filtration, you can't really do 100% medium exchange. So, but with this particular vessel, because of the positioning of the sampling ports, so it's all due to design of the vessel essentially. So because of the positioning of the liquid removal port, because actually I think that's how it's officially called, you will always have a dead volume of approximately 500 milliliters, unless you want to use the really bottom port, which is for harvesting. But then that means that you would, if you only want to do a medium exchange, you will remove all of your cells. So you don't want that. So the only option that we have with this particular bioreactor vessels, there are others which have different, that will allow you to perform a higher, remove a higher volume, but with this one, it's only 50% if you're running the bioreactor the 1 liter scale. So these are the parameters that we've used initially. And this was the first couple of runs that we've done. And you can see that there was quite a nice growth. So we've put in 7.5 million cells based on that seeding density and surface area. And we got out around 300 million cells, which was pretty good. But we know we can do better. And these are actually some images of live dead staining. So live cells appear as green, dead cells appear as red. You can see some here in the bottom image at different time points in culture. Yeah. So you can see at day 10, the level of aggregation was actually quite significant, quite increased. Right, so we know we can do that better than this. So what we did was we went back to those latest experiments with the microcarrier and the volume addition. We've used the same time point for doing the steps, so day 5. So essentially now at day 5, we went from 5,000 square centimeters in the 1 liter to 10,000 square centimeters in the 2 liter volume. And what we've seen was quite an impressive cell growth. So if you look at the numbers, if in the baseline, which I've showed in the earlier slide as well, we've got around 300 million cells. Well, now we've got around between 800 and 900 million cells, and we've put in exactly the same amount, which was 7.5 million. So in a fold increase, that translates to the baseline was close to 50 fold, so I think it was 47 fold, while for the microcarrier and volume addition, it was over 110 fold increase in cell number. So that was really, really exciting for us. So as usual, we looked at glucose and lactate. So the top graph is for the baseline. And we don't really see depletion of glucose here or accumulation of lactate. Lactate towards the end of the culture only reaches values around 4 millimole per liter, which was still all right. But for the microcarrier and volume addition, we actually see, particularly towards the end of the culture, glucose is very, very low. We managed to increase it with the last medium exchange, but the next day was very, very low again. Okay, so almost depleted. With lactate, that has increased. It was between 6 millimolar and 9 millimolar per liter. So that is quite a high concentration. As I said, the inhibitory, well, the concentrations of metabolites that will determine growth of rest are different for each species. We don't really know for bovine, but it is quite a high concentration. Another thing that we've also looked at and we've monitored in these bioreactor liter scale runs was the pH, which was pretty similar for both baseline and the microcarrier and volume addition. And it was pretty easy to maintain it between the values that we've set up without actually adding anything to it like acid or base. We could control it simply through the medium exchanges. We also monitored dissolved oxygen. So gassing in the liter scale bioreactors, we've decided to do it by using the headspace aeration. And we've only introduced air and CO2, so carbon dioxide. We haven't introduced any oxygen whatsoever. Not in these runs, but what you can notice towards the end of the culture where we know the numbers are really, really high at that stage is that dissolved oxygen goes very, very low. I think in the last day it was something like 1.5% dissolved oxygen, so that is very low. There are arguments here that we don't really know if this is good or bad, given from the lessons that we've learned with human cells, human mesenchymal stem cells. But I think it's definitely something that we need to keep an eye on. So next steps and things that we also want to investigate is essentially to see if we can push this even further. If we can get over 1 billion cells by putting in less than, well, 7.5 million. And I think the next runs that we have planned are to now switch, because when we're running the bioreactor to liters, we have the capability of even taking into consideration that 500 mils of dead volume, we have the capability of doing up to 75% medium exchange. So the next runs will look at that. After day 5, when we double the volume, we're going to remove 1.5 liters out of the 2 liters, which will give about 75%, and see how that affects cell growth. And also, we also want to, we're also continuously working on developing that optimal differentiation media formulation, not just for fats, but we're also looking at the muscle differentiation. And the next steps would be to, once we have a formulation that we're happy with, to integrate that into a scalable bioprocess. So go back into the spinner flasks first, use that and try to find all those optimal parameters for differentiating the cells. And then obviously at the end, we don't have much time on the project left, but maybe this will be a continuation of the project to actually start looking into co-culture of cells, and maybe even encapsulating the cells so that you can give them that 3D environment where the cells can interact with each other to form the meat, the microtissue. So obviously I would like to thank Good Food Institute, because without you, and without your funding, this would not be possible. These are some images of some of the people in our cell technologies group and users of our lab MB334. And then in the bottom picture, you can see Panagiota Mutsatsu, who initially she was meant to be the postdoctoral researcher on this project, but Mozambique recruited her before before she got a chance to start on the project. So that was really exciting for her, because obviously she's moved on to really exciting stuff. And then we've recruited Dr. Junaid Ali, who in May left, and he went to work for Gourmet, which again is a cultivated meat cultivated foie gras company based in Paris. And then Fritz de la Raga is, well, he used to be my PhD student, and now he also works on the project. So thank you so much to Fritz. Thank you so much to my mentor, who, sadly passed away last year, Professor Chris Hewitt. And my other mentors, Professor Alvenino and Prof. Ivan Wall, who now I work with really, really closely, and everyone in my lab, and everyone for listening. Thank you.
Thank you so much, Petra. I really couldn't thank you enough. That was such a phenomenal lecture. Specifically, I loved how detailed you were able to get in terms of all of the optimal parameters you deduced, in addition to also keeping information really accessible. It felt like kind of a course in effective biological engineering or bioprocess engineering. So thank you so much for sharing this information with us. I'm excited to watch it back 2 or 3 times over once the recording is up on our YouTube channel. I am going to ask you to stop sharing your screen now, and then we'll move to the Q&A portion. Lots of folks in the chat that are really enthusiastic about your lecture, and who are also really excited and grateful that you've been publishing your findings in an open access manner. Because yeah, we definitely—
hopefully there will be another one on this latest work that we've shown. So we're definitely preparing the manuscript now, but most likely it's going to come out next year for sure. So
awesome. We'll be very excited to follow your work. So let's move to the Q&A. We have roughly 20 minutes or so left. You've been speaking for a very long time. So
oh, no, sorry. I think it's a
oh no, sorry. That wasn't
professional defect. Like, it did feel a little bit like a lecture. So
It was really good, very thorough. But I just mean that if you need to take a break at any point, do let us
Oh,
know.
that's all right. That's all right. I'm actually very excited to hear the questions and what the audience, what they think, and maybe they can come up with new ideas. Maybe they know more about adipogenic differentiation, which is something that we're trying to find out more now and investigate. So, yeah, any suggestions, anything, it's very welcome.
Perfect. Thank you, Petra. Everyone, if you have questions for Petra, please do drop them in the Q&A section in your bottom bar. And I'll start moving through some of the questions that we've received. Yeah, lots of folks expressing enthusiasm, saying that this was a really phenomenal lecture. So the first question that I think is quite interesting is from Maya Banami. Why recreate the wheel if there exists more successful research on adipogenesis in human cells? Is it a taste or ethical concern or a kind of compatibility issue to mix human adipose cells with animal muscle or collagen cells?
I mean, so I think it's a really interesting question. To be honest, I'm kind of trying to wrap my head around it on how to answer this. But our thinking about cultivated meat is that it's very complex tissue that doesn't just have one cell type in there. It doesn't just have muscle. It also has fat. I mean, obviously, it depends on what kind of cut of meat you're looking for to produce, because the concentrations of, oh, sorry, am I still sharing slides? No, okay. So the concentration of fat that is, the percentage of fat that is in there is going to be different in every single cut of meat. But I think you do need fat. And if you think about it, if you only have a piece of meat that is made of muscle, it will not have the same nutritional profile as meat. It would not have the same taste, as meat. So I think fat is pretty important.
Yeah, thank you. I would agree with that. I'm not not terribly interested in eating a pure sheet of muscle. Here's a question from one
Easier to make, for sure. So it
right.
probably be easier to make, but if we really want to have all those benefits from cultivated meat and take care of all the environmental issues, or at least address a proportion of those, I think we need to have cultivated meat products on the market that will mimic the animal meat as closely as possible. And that includes not just the texture, but also the taste and the profile, the nutritional profile. And I think with cultivated meat, you also get this advantage that because you're producing it in a controlled manner, you can actually have flexibility of improving that nutritional profile. Maybe, I'm not sure if the technology exists, maybe I'm a dreamer, but it could be that maybe we could add omega-3 oils, which we know they're really good, but you normally find those in fish, don't you? You don't find them in normal meat. So maybe you could add those in there, or maybe you could add other things that can help,
Absolutely.
alongside the texture and the taste.
Agreed. That kind of potential to maybe expand the human palate with brand new meat products, but also B, to solve some like population health issues and potentially close health disparities, without preventing certain subsets of the population from consuming meat products. Yeah, lots of potential there. There's some more technical questions that are coming through that I'd love to get your thoughts on. So from Varsha, how do you translate the decreased CFU observed at passage 7 to what would happen on microcarriers without the repeated trypsinization?
That is a really good question, Varsha. So I think if you start with cells which are pretty old, like passage 7, we found out that they slow down their growth, and you put those on the microcarriers, you would probably not see a lot of cell growth in the first instance. But if you start with early cells and you repeat, so I mean, the way I'm interpreting this question, when you say what would happen on microcarriers without the repeated trypsinization, the way I'm seeing that is that probably what I've done with the microcarrier addition, you're thinking of doing that several times so that you can keep on boosting the cell number. I'm guessing that's what at least that's how I'm interpreting it, without essentially trypsinizing the cells, seeding them into a new vessel, etc. So I think you have to bear in mind, the longer you culture the cells, regardless that you give them more surface area or you don't, the longer you culture them, if they like it, if all the conditions are right, the cells will continue growing. So by growing, it means that you will get higher and higher population doublings. With these cells, because they're primary cells, you have to bear in mind that the number of those population doublings, regardless of how you culture them, you're still going to have a maximum limit. So once they reach that limit, then they're not going to grow beyond that. Yeah. So regardless of what you do in your bioprocess, if you carry on the culture for very long, you will most likely reach that limit for sure.
Thank you, Petra. Such a thorough answer. Here's a question from Shira, just a kind of an admin question. Is the insulin that you used for the preconditioning, is it of bovine origin?
Yeah. So insulin, this was one of the easiest ones to find. And it was from bovine pancreas. So yeah.
Thank you. Here's another question. Did you measure the adipogenic differentiation efficiency? Sorry, how did you measure the differentiation efficiency?
Yeah, so it was done by image analysis. So that was the reason why we've chosen Lipitor, which is a fluorescent stain, so the green that you've seen in those images, and we've combined that with DAPI. So what you do, what you get when you image them, you have images for each channel. Yeah, so DAPI would be blue, Lipitor would be green, and then you have your merged image. And by using something as simple as ImageJ, which is it's a free software, easy to download from the internet. What you can do is you can manually count. I mean, if you're really good with technology, you can actually create a macros and use that to make your counting automatic. I used to do it manually. So you can manually count the number of nuclei that you have in your image. That will give you the total number of cells, and then manually you have to pinpoint those cells that have Lipitor in your merged image. And essentially that's how you calculate your adipogenic differentiation efficiency. You know how many of those total cell number have formed those adipose vesicles, fat vesicles, which essentially are typical to adipocytes.
Wow, that is wild that you used to manually count the cells.
I know. I know, but what do you do for science?
Yeah, what we will do to get to the truth. That's too funny. Here's a question from Ted. Did you test myogenic differentiation ability of the bASCs? If not, why?
So this is, oh, sorry, go ahead. Yeah. So we're still working on that. As I said, we're testing a range of media formulations now, because obviously that was the initial reason why we chose these cells, even if initially we were aiming for bone marrow derived bovine MSCs. So from the studies that we found, which look at different media formulations for myogenic differentiation, they all take at least 6 weeks. So we're kind of in the process. It's a very, very long process just to carry on. Essentially, you're just doing medium changes until your 6 weeks timeline, when you fix your cells and you get to stain them to see if there was any change. So we're still in the process of doing that. If COVID didn't happen, we would have probably be a lot more ahead with that. But unfortunately, yeah, we are where we are. But we are testing several, well, we're kind of doing multiple things in parallel to try and get as much data, as possible. But yeah, we're still working on that.
Yeah, thank you. Lots of unsolicited rest and recovery time for our researchers this season. Um, yeah, so here's another question from Bezalel. In culturing the cells, do hydrocolloids play any role? And if so, have you explored the effect of clean label hydrocolloids on the cell culture?
Ooh, so that is a really good one, because first of all, I'm trying to think. So I'm trying to think what exactly you're referring to when you talk about hydrocolloids. Colloids. I'm assuming components in the media. So we're not really adding anything additional to the media than you would normally do. We're not really putting in anything rather than, I don't know, maybe small molecules, FPS. But yeah, I'm
Yeah,
not really sure.
no, I
To
think
be
that helps.
honest.
It's I think they're referring, I had to look this up as well, but like polysaccharides of that are extracted from plants and algae. That are,
Okay.
yeah, I I don't know the answer to this, but I think it's maybe an interesting line of
Yeah. I mean,
exploration.
to be honest, I don't know the answer to this either, but I think it's definitely something that, we can look into. Maybe those hydrocolloids could actually help with developing some sort of a serum-free media to replace different things.
Yeah, and so
No idea. Sorry, Bezalo. I don't know.
no worries, that's great. So here's a question from Soren. I wonder how we can kind of overcome the issue when it comes to scale up with only 7 passages. Like, how can we produce seed inoculum at the final stage?
Yeah, that is a really well, that is a very big challenge. But I guess one way of doing it would be to immortalize these cells, and then you don't have that limitation anymore. But we do know that, by immortalizing cell lines or primary cells into, immortal cell lines, you are essentially changing the cells completely. Yeah, surely you're giving them that, that potential of proliferation, extended proliferation, unlimited almost, but then those cells will modify, they will adapt to that sort of culture and modify to the stage that they're not really resembling the initial tissue anymore, which could probably be a good thing. I mean, thinking about CHO cells, because those are the most common example of, of that sort of cell line that has been modified and adapted to the point that now you can grow them in 10,000 liter bioreactors to produce proteins and things. Who knows? Maybe, I mean, I'm not aware of anyone trying to do that with stem cells or modify them, adapt them to that stage. But I think if you can do it safely in a way that you can still essentially maintain those quality of the cells, like their differentiation potential, because essentially that's what's of interest here, isn't it? The differentiation potential towards those cell types of interest. Then I think that would be ideal, because then you would have no limitation whatsoever. Your cells would grow, will proliferate forever, and you could put them in those 10,000 liter bioreactors, and they will be really robust. So yeah, definitely something to aim for, I think.
Yeah, absolutely. We have 7 minutes left in our kind of allotted seminar time, and 14 questions left to go. I think
Oh no.
realistically, we're not going to get to all of them, which is totally okay. Maybe folks can reach out to you via LinkedIn if they would like to have some further conversation and ask more questions. As you scan through this question list, if there are any that kind of call out to you that you especially want to answer, feel free, but I will continue kind of asking the ones that I am interested in.
Yeah, of
There's
course.
so many good questions here.
Go for it.
Yeah, so Aditya asked, he's another one of our student leaders, what are your thoughts on the potential applications of metabolic models of MSCs in optimization of media for growth and differentiation?
Ooh, so I think that would be a really good one if we can optimize the media to the point that it becomes really, really cost effective. Obviously, that is something to look forward. And I mean, there's so much that we don't really know or we can't really measure in the media itself. So when we change, I mean, as I said, what we do in my lab, we just monitor glucose and lactate, but that's not enough. I mean, that's probably 0.00001% of the important things that you can find in the media. So actually being able to measure, you know, if you have that capability of metabolomics and measure everything that you have in your media at the different time points in culture, that sort of data can inform your medium, your feeding strategy, essentially. So, you know, I did say 80% was the best for us in terms of the cell number, but by actually being able to control that down to the molecular level, I think that would be fantastic, because that could save a lot of money.
Absolutely. Thank you. Let's see, maybe we'll strive for 2 or 3 more questions here, last few questions. There are a lot of questions about the microcarriers. So maybe we'll ask this one.
Okay.
What materials were used for the microcarriers, and is this method food safe?
So these microcarriers that we used are commercially available ones. They're polystyrene, and the reason why we chose these for the initial bioprocess development was because polystyrene essentially mimics tissue culture plastic. And we know they've worked really well with human cells, but obviously to get cultivated meat as a product, you want to replace even these microcarriers with something that is most likely, well, ideally edible, right? Or something that can be broken down into components which are not toxic, or they wouldn't cause any sort of reaction. So the microcarriers themselves, I didn't produce them, I just bought them made. But I know there are various groups that are actually working on edible microcarriers, which is really, really exciting. And, you know, fingers crossed, we're going to get something available on the market, because I would really, really love to try those in my bioreactors.
Awesome. Maybe one last question here. We have some time. We can squeeze in 2, I think. A question here from Anne Sophie, who said that you gave a really awesome lecture and is curious more about the more downstream elements of the bioprocess. So once these cells are proliferated, what are the steps that you need to differentiate those cells in the bioreactor? And how are you planning on detaching the cells from the microcarriers that you're currently using, or using edible microcarriers like you just kind of discussed is a workaround for that?
So harvesting cells from polystyrene microcarriers obviously will need enzymatic treatment. So we have protocols that we have developed in-house, and they work really, really well with human mesenchymal stem cells. We've tried them with bovine, and they've also worked really well, because at the end of the, you know, each of the spinner flasks and the bioreactor cultures, we harvested the cells so that we can essentially seed them for characterization. So we had to go through those steps. I can tell you practically, it's a very lengthy process to harvest the bioreactor. It doesn't matter, liter scale or 2 liter, well, actually 2 liter scales is slightly more, it takes slightly longer, but it takes somewhere around 6 hours. And the reason for that is because you have to do all these steps. So think of it like how you would do, we actually have a paper published on that as well, on harvesting protocol from polystyrene microcarriers, but that was for human cells. So as I said, everything kind of comes from our experience with human cells, and, you know, we've just adapted it to the bovine cells. But essentially think of it like the procedure is identical to what you would do in monolayer, in culture vessels. You have to remove your medium, you have to wash your cells with PBS so that you can remove you know, any traces of that serum, because serum contains trypsin inhibitors. So if you don't remove the serum and you add your enzyme in there, it's not going to work. But the difference in bioreactors is that you do that under agitation, because If you remember some of those images that I've shown, you can get towards the end of the culture, you can get very high levels of aggregation. So breaking those clumps of cells and microcarriers down require quite a bit of force. You can't really do that in static conditions. You need that agitation, and it's actually quite increased agitation, so that you can release the cells, and you can get the enzyme to actually get into the cores of those aggregates as well. Yeah, because some of them, they were pretty big. So at the moment, we're doing that by obviously using proteolytic enzymes. It doesn't seem to affect cell quality, at least not what we are measuring. But then again, there's a bunch of other stuff that we could measure to look at the quality of the cells that we don't really know how it impacts. So yeah, ideally, surely you would want something that you can just disintegrate and it will release the cells. So like an edible microcarrier or dissolvable microcarrier, that you can do that by, I don't know, maybe something like using a chelator agent, DTA, to take those ions or something like that. That would be really, really useful.
Awesome. Thank you so much, Petra. We are just about out of time. I want to give just a massive, massive thank you to you again. I mean, right out of the gates in this lecture, when you had that slide about the advantages and disadvantages of each of the different cell types from embryonic to iPSCs and all of that, like even that alone was just like so incredibly valuable to our field. And yeah, you've only continued to wow us, I think, with your knowledge, and then also your openness to sharing that that knowledge for the betterment of this field. So very much appreciate you. I would love to turn it over to you for any final thoughts you want to leave this community with. And then we'll kind of wrap up and say, see you next month to the rest of these scientists.
Yeah, well, thank you so much, Amy. Thank you so much, everyone, for joining me. I hope you enjoyed my talk. If you do want, because I know there have been a few questions that haven't been addressed, so if you do want to get in touch with me, my email is m.hanga@aston.ac.uk. You can also find me on Twitter or LinkedIn, so definitely get in touch if we didn't get around. And yeah, I'm really, really excited to see, this community growing and having so many more people interested in working this area. I started working in this, a year and a half ago, and there weren't really many people to get in touch with and talk about these things. So now, through all the hard work of GFI, it's pretty unbelievable, and there's a huge community now. So I'm really, really grateful to GFI and everyone.
Thank you. We're so grateful to you as well. All right. Thank you, everyone. Thank you, Petra, again. Thank you, everyone, for joining us today. Be sure to check in again next month, where we'll have another GFI grantee, Dr. Amanda Stiles, come in and give a presentation about red seaweed protein. So really looking forward to seeing everyone there. Thank you so much for being here, Petra. We are so grateful to you and everything you do for our field. And I hope everyone has a fantastic rest of your week. Thank you.